Well, welcome everyone. I'm Joanne Geer. I'm Executive Director of the Biopharma Research Council, and welcome to our webinar on the second harmonic generation, imaging and quantification of fibrosis of the liver. I'll be telling you about our speakers in a moment, but first I'm going to just tell you a little bit about us. We are an association, well, there you go. We are an association of there we are at the beginning. We're an association of scientists throughout all the different silos of biomedical research. And we use scientists and researcher broadly. We include engineers, data scientists, and so on. Our whole mission is to stimulate collaborations and partnerships that can accelerate medical progress. I'm just going to show you a few of the things that we have planned this year. We have quite a few different programs. All of these are on our website, and I'd be happy to chat with anyone who'd like to know more about them. But just to see, we've got a big year ahead of us, and we're very happy that you're with us. The big event is we're doing a manufacturing expo in New Jersey in the fall. This is a new thing for us. In today's webinar, you can enter questions in the question box at any time. If it's about a specific slide, you can feel free to be specific about that. We're going to hear from both of our speakers, and then we'll open the Q&A time. So about our presenters today. Dr. Petitjean is an experienced leader with 20 years of executive level and international business experience in medical technology and life sciences, including 12 years at General Electric Healthcare in both product and business leadership positions. He has extensively published and created intellectual property in the areas of molecular beam epitaxy, semiconductor bandgap engineering, and medical cellular imaging. Dr. Li Shen, formerly with ImClone and Hoffmann La Roche and Zeus Scientific, is an experienced pharmaceutical research scientist familiar with most in vivo and in vitro techniques and related workflows used in industry. More specifically, she is experienced in assay development and validation, demanding regulatory and quality assurance frameworks, related technology transfers, and management of programs with CROs or technology vendors. Dr. Petitjean.
Hello, Joanne. Thank you so much for welcoming us, and thank you for the Biopharma Research Council for inviting us today for this presentation. Indeed, we are all aware that there is an increasing demand in the area of fibrosis, and in particular in the development of anti-fibrotic fibrosis treatments in the US. And as a result, quantification of fibrosis becomes an increasing area of interest. Let me tell you about our venture. We have created, in partnership with HistoIndex, a very visible startup out of Singapore who has developed the equipment that we're using, a imaging lab here in Princeton. And our goal is to provide very robust and translational methods to quantify fibrosis. We are using the method developed in Singapore based on second harmonic generation and 2-photon emission, and I will explain how this works, as well as validated machine learning image analysis and quantification algorithms. And the workflow has been designed to be extremely friendly with existing workflows. I want to say that we'd start with conventional non-stained pathology slides, we image them, and then we quantify the data. And for now, in order to give access to the industry, to this technology, we have adopted a pay-per-use, pay-per-study model, and additional options to access the technologies are described on our website. So now that I've explained the overall mission of our lab, let me tell you how this works. Second, nonlinear optics, it's not a new science. What I'm going to show you has been extensively published. The new thing is that we can have access to all this technology into tabletop systems as the Genesis system provides. And in that system, we basically have access to 2 optical methods. The first method is 2-photon autofluorescence. So basically what happens, we use 2 consecutive photons in the red spectrum to really activate proteins, intracellular proteins, and those proteins will exhibit autofluorescence in the ultraviolet in the blue area. So 2 photons in the red equate to 1 photon in the blue, and this is when we have high energies to activate, for instance, NADPH autofluorescence. We don't use any biomarker, optical biomarker. So that's one process. The images coming from fluorescence will show in red in all the images that you're going to see in the rest of this presentation. We also use second harmonic generation. Second harmonic generation is another optical method that is sensitive to non-centrosymmetric molecules such as collagen, microtubules, and others. And we have optimized and tuned our equipment in a way that second harmonic generation is specific to collagen 1 and collagen 3. And the related images will show in green in all the images that we'll show today. And then we overlap both. We colocalize the extracellular information from second harmonic generation and fibrosis with the 2PE image, which gives us context and intracellular information, and we generate those images. Just to give you a quick overview of how this shows. So here you have a typical image of a non-stained tissue. In green comes the collagen and the fibrosis, and in red would be the extracellular, the cellular information. The red comes from NADPH, NADH autofluorescence, and the brighter the red, the healthier the cell. To the left here is the corresponding picture with conventional Picosaurus red stains, both at 20x. The areas don't really match exactly, but you have here an overview of how these images compare. So the images we obtain look like this, right? And this is a zoomed in image. So this is a typical NASH model. In green are the collagen fibers, right? And you see them with different intensities. The more intense, the more fibrils you have in that fiber. And in red is the tissue. We can see here the fat, but there's absolutely no signal. And when we eliminate the red channel, these are the kinds of collagen images that we obtain. And as you can see, they are extremely rich, and we can detect many, many levels of collagen, which makes this technology and this approach extremely sensitive. Here you can see a full image of the liver, and we are focusing on that little red field of view and region of interest. So this is just to train you on the images that you will see later on in this presentation. So we can image many, many things. The system has been tuned to image collagen and autofluorescence from NADH and NADPH. And in addition to that, we really focus on optimizing the image quantification on fibrosis-based concerns. So once we image tissues, and this is the list of tissues that we are working on, we have developed a very structured method to quantify those images. These images, the way we quantify fibrosis is based on a machine learning process. I will explain in detail if there are questions. But in short, we teach the software to recognize and then to quantify features of fibrosis, and pathologists train the software. The process takes 3 steps. First, we explore how to do it. Second, we develop the algorithms. Third, they are validated into trials, and I'll show you some publication, and then they go commercial. At this stage, we are commercial in liver, kidney, and lung. Today, Lee will show you how it works in liver, and we are currently developing this entire funnel of applications. It takes about 12 to 18 months to develop and validate an application. Let me quote 3 papers that have been published. You can see the papers on our website, or I'll be happy to send them to you. They're all on liver. The first one is a typical mouse CCL4 model, and the goal of this paper is to introduce one of the very first quantification index, which we call the fibrosis index. We have compared that index to histopathological score, and we see that our index progresses very nicely in comparison to the histopathological score. And the goal of this study is really to show how our method minimizes intra and inter observed discrepancies, provides a standard way of indexing the tissues, and also significantly reduces the variability of the measure because we eliminate tissue processing and tissue staining. To the left, you have a few comparative images between our technique and H&E. The second paper that I would like to quote also refers to liver and introduces and explains the Q-fibrosis. The Q-fibrosis is another quantification index that is much more sophisticated, and we benchmark against metavir fibrosis stages. The study compares and discusses 2 aspects, a preclinical aspect and a clinical aspect with human tissues of patients affected by hepatitis B. And the way the study is run is really basically after we harvest the tissues are split between the conventional scoring method through a microscopy and staining and histological assessment. And the second is our unstained method. We do nonlinear optics, and we quantify using the Q-fibrosis. And as you can see here, both for the animal and TAA model and for the human, our scoring method compares extremely well and has a much more dynamic range than other methods, and compares as well to the total collagen content that is done. The last paper that I'd like to quote introduces advanced quantification. So again, just to rewind a little bit, the first paper talks about our basic quantification. The second paper talks about our quantification system as it compares between animal and clinical. And this paper introduces the capabilities that we have to do really advanced quantification of fibrosis, and in particular fibrillar collagen. So here is it's a human study. Here are typical examples of the way we segment the fibrosis and how we focus on those little fibers that you see here, that are reported here. And we have the capability to really analyze each little individual fiber in many morphological dimensions. So, you know, size, brightness, distribution angles, all kinds of parameters can be described. And the advanced quantification score utilizes 13 different morphological scores. And when we compare this method in red to the basic quantification, and this is based on total quantification collagen levels through histopathology assessments, we show that the advanced quantifications gives us much more dynamic range, in particular in the areas of early fibrosis progression. So this is basically what the study concludes, is that morphological features such as collagen string formation and connectivity may allow us to identify several stages in the progression of fibrillar collagen formation. I use those 3 examples to show you how the technology is translational. We can use it from research to the clinic. The research system is a mid-throughput 10 slides per batch system. The clinic system is a 1-slide system. I want to say that for now, those systems are used for research only. but we are looking at promoting this technology for clinical applications in a couple of years. And as I said, we have 3 commercial applications: liver, lung, and kidney. And today, Lee is really going to show you several case studies on liver and really how we run this technology for clinical trials. Thank you so much for your attention. Now let's turn to Lee's slide deck.
Thank you so much, Matt. So hello, everyone. Today I'm going to be presenting to you 2 case studies. The first is going to be the carbon tetrachloride-induced liver fibrosis study in mouse, and then the second one, less in-depth, will be for the NASH study with fatty diet-induced liver fibrosis. So to start out with, I want to give a overview of our capability for tissues and then our collagen quantification parameters. So on the images you see here, the green images is from second harmonic generation for collagen. The red image you see here is from the 2-photon excitation, the autofluorescence from the tissue to look at the tissue morphology. And these tissues, they are actually FFPE, formalin fixed paraffin embedded tissue. They're about 5 micron thick. Optimal is 5 to 10 micron thick, and we received them non-stained. So after we image the image and then we do collagen image analysis, for the basic quantification, we have the percentage total collagen. Percentage total collagen can then further be divided into the distributed and aggregated collagen. For advanced quantification, we have the collagen segmentation into portal, septal, and the parasinusoidal region. We can also quantitate the collagen fiber, morphometry, and frequency, and also the reticulation nodes. So for the first case study, carbon tetrachloride-induced liver fibrosis, this is the study design. So this is a very small subset of samples from a much larger study. So the sponsor gave us 10 samples. There's 5 groups, and there's 2 animals per group. We were first blinded to the treatment group. We only provide the sample ID, but now we know the treatment group. So the carbon tetrachloride, they're in olive oil mixture. So the group 1 is olive oil control. Group 2 is carbon tetrachloride at 2 weeks. Group 3 at 4 weeks. Group 4 at 6 weeks. And group 5 at 6 weeks with drug treatment. The drug they use is pirfenidone. Pirfenidone is an anti-fibrotic drug for treatment of idiopathic pulmonary fibrosis. It was first approved outside of the U.S. On the right here, you see is the non-stained slides that were provided to us. You see that the tissue here is quite large, so it gives us a very nice sampling area for imaging and image analysis. So right now we're going to go into some of the images that we obtained from our technology. So this is a control sample. So the red color you see here is from second harmonic. No, so the red color you see here is from 2-photon excitation. They are autofluorescent from the tissue to look at the tissue morphology. The green color you see here, that's around the border of the portal and the central vein, is from the second harmonic generation for collagen. So this is a control sample. It looks very normal. If I remove the red channel, you'll be able to see the collagen. There's a lot of small collagen fiber spread throughout the tissue. So to compare to this, I'll show you a more advanced fibrosis. This is carbon tetrachloride at 6 weeks. You see here it looks very different from the control. You see that there's elevated collagen deposition and also fibrotic septa. You see very thin collagen bridge here, as well as really widened fibrotic septa. And this looks like a barbed wire here. It's spread throughout the whole tissue. And if I remove the red channel, you see the collagen much better, and you see that it's everywhere. The collagen, this elevated collagen deposition and fibrotic septa. And if I bring the red channel back, you'll be able to see a lot of interesting features. You see a bright red hepatocyte along the fibrotic septa. And so we think that these are activated hepatocyte. We know that there's also activated macrophages along the fibrotic septa. But these cells are pretty large, so it looks like hepatocytes. And then another interesting feature from these carbon tetrachloride treated animals are these, this is from another sample, are these glob of collagen, and within it there's conglomerated cells. They look like hepatocytes or activated macrophages within this glob of hepatocyte, glob of collagen. So going back into the presentation. So now we're able to compare between the different treatment. So there is 5 groups in the study. There's 2 animals per group. You see here the top row is animal 1, the bottom one is animal 2. We're looking at control with olive oil alone. 2 weeks, carbon tetrachloride, 4 weeks, carbon tetrachloride, and then 6 weeks, carbon tetrachloride, and 6 weeks carbon tetrachloride with drug treatment, pirfenidone. I'm going back to the original slide. You see here that in control animal, there is very little collagen deposition. By 2 weeks with carbon tetrachloride, you see a thin bridge forming. The fibrotic septa is very short. They're very thin, coming from the portal. And then by 4 weeks, this fibrotic septa has expanded itself, has extended more into the tissue. There's animal variability in this study. This animal here at 2 weeks has advanced fibrosis. So if I remove the red channel and just look at the green channel, you see the collagen much better. You see that there's elevated collagen deposition at 2 and 4 weeks, and you see the thin fibrotic bridge here, and a little bit more advanced at 2 weeks for this animal. So now we're going to 6 weeks. So at 6 weeks, by 6 weeks, you see that there's massive collagen deposition. You see fibrotic septa forming. You see very thin fibrotic bridge. You see barbed wire-like extension of the collagen bridge, and this extension has reduced with drug treatment, pirfenidone. You still see the thin bridge with pirfenidone, but the barbed wire collagen septa has disappeared, well, at least reduced. And you can see much clearer when you move the red channel and just looking at the green channel. The thin fibrotic bridge is still here, but the barbed wire has reduced. So now we're going to collagen quantification data. So this is total percentage collagen. We are looking at control, 2 weeks carbon tetrachloride, 4 weeks, 6 weeks, and 6 weeks with drug treatment, pirfenidone. You see here that there's elevated collagen deposition at 2 weeks, and this increase continues at 6 weeks and peaks at 6 weeks. There's a reduction of the collagen with drug treatment. So now we're going to, so same thing, this is the total collagen I just showed, but total collagen is made up of aggregated plus the distributed. Now I'll define the aggregated and distributed collagen. So collagen fiber can be by itself, not touching each other, as in diagram A. The collagen fiber can be in a simple cross, as in B. Diagram C shows a simple cross with one run-in fiber, or D in plate formation, it looks like a braid, or E, S run-in fiber crossing through two parallel fiber. So diagram A is the distributed collagen. Diagram B to E is what we call aggregated collagen. So when the collagen fiber cross, the point that it crosses is the reticulation nodes. The more the collagen, the more complex the network of collagen, then the more reticulation nodes you'll see. So on the right here is an image that we typically generate. So the yellow color you see here is a hand-free draw into the collagen fiber. You see two collagen fiber crossing. The point across is the reticulation node. Going back to the collagen quantification data, you see that the aggregated collagen here is the results very similar to the total percentage collagen. And for the distributed collagen, you see that the elevation of the collagen is mostly at the 6 weeks. 2 to 4 weeks is a very similar level compared to control. This elevation at 6 weeks is reduced with drug treatment or distributed collagen. Okay, so now we're going to collagen segmentation for 3 different regions: the portal, septal, and perisinusoidal. What we call portal collagen is the collagen around the boundary of the portal and the central vein. You see here in green. The septal collagen is the collagen between the portal tracts and the central vein. It can be a complete bridge, as you see here, as in this case with carbon tetrachloride treated animal, or it could be scatter. So these are mostly collagen fiber in the perisinusoidal area between 2 portals. What we segment into the perisinusoidal collagen is the collagen in the perisinusoidal space of Dicey. They're the collagen that's not segmented into the portal nor the septal. So, so this is collagen segmentation images. So this on the left here is the raw image, and then these are segmented into portal, septal, and perisinusoidal. The portal image you see here has the boundary of the portal and the central vein. So it's segmented very nicely here. The septal collagen is the collagen between the portals and the central vein. You see all the septal bridge, the collagen bridge here. And then the perisinusoidal image has the collagen in the perisinusoidal area that's not in the portal nor the septal. They're usually very small collagen fiber. So, and because these are very zoomed out image, this is a very large piece of image. You have to really zoom in order to see these small collagen fiber. So this small image here is a zoom in version. Okay. So this is another example of the collagen segmentation into portal, septal, and perisinusoidal. So on the left here is the raw image. And right here, the portal, you see here, it's segmented very nicely in the portal collagen. And then there's a bunch of small portals here. And then these are the collagen bridge in the septal area. And this is the perisinusoidal. So now we're going to the collagen segmentation into the 3 different regions, the collagen quantification values. So on the left here is total, which I show in the previous slide, and then segment into portal, septal, and perisinusoidal. You see here there's the elevation in collagen deposition in the portal area by 2 weeks, and it peaks at 6 weeks. There's a reduction with drug. For the septal, there's a very nice elevation of collagen deposition going from 2 weeks to 6 weeks, and a reduction with drug with pirfenidone. For the perisinusoidal area, there's it pretty much stay level at 2 and 4 weeks with a elevation at very visibly seen at 6 weeks, and then a reduction with drug. Okay, so next we're going to a collagen fiber content. So I want to give you show you this very simple diagram. Yeah. As you know, that the collagen, the basic unit of collagen is the triple helix. The triple helix, they self-assemble and cross-link to form the collagen fibril. Collagen fibril is about 10 to 300 nanometer, and these collagen fibril can then bundle into a collagen fiber. Collagen fiber is about 0.5 to 3 microns. So on the right here, you see is the electron microscopy image, and these are collagen fibril. The diameter of this collagen fibril is about 50 to 75 nanometer. So electron microscopy can image at the level of collagen fibril. For us, Genesis Image, we can image at the level of collagen fiber. Our resolution is about 1 by 1 micron. So these collagen lines you see here are collagen fiber, and we sometimes call this collagen strings. So next, we're going to total collagen, number of total collagen fiber and collagen segmentation into the 3 regions, the portal, septal, and perisinusoidal. So looking at the total number of collagen fiber, you see here that there's a high amount of collagen fiber in control, and then maybe even a slight reduction at 2 and 4 weeks, and then it elevates at 6 weeks with a reduction with drug pirfenidone. And so I'm going to go more into this. So the reason why in controls there's high number of collagen fiber is because they're mostly very small collagen fiber. So if we go back to the original image here, so this is the control image. So if I remove the red channel and we zoom in, you see that there's a lot of small collagen fiber here. So although in control there's a very small amount of percentage total collagen area, but if you're counting collagen fiber, which is another way to quantitate collagen, you'll see that there's a lot of small collagen fiber here, and this adds up to a high number of collagen fiber. And then if we go back to the quantitation data for the distributed collagen, you see here even a control compared to 2 and 4 weeks, there's high amounts of distributed collagen. The distributed collagen are the fine collagen fiber that's by itself, they're not connected to other collagen fiber. And then, and lastly, in the next slide, when I talk about collagen fiber morphometry, you see that there's a lot of small, that we count a lot of small collagen fiber. I'll show that in the next slide. But let me finish on this slide. So when the number of collagen fiber are segmented to portal, you see that there's elevated number of collagen fiber in the portal area at 2 and 6 weeks, and with an increase with the drug. For septal, you see that the elevation is at 6 weeks, and it stays up with drug. For the perisinusoidal area, you see a very nice step-ladder increase from 2 to 6 weeks and peaks at 6 weeks, and it maintains with drug. So now we're going to collagen fiber morphometry, where we're looking at the number of short strings, long strings, thin strings, and thick strings. The total number of strings is actually made up of the short plus long, or thin plus thick. So, as we have seen before, that there's actually a, so the control, there's high amount of short strings here, as I talked about, and also a high number of thick strings here for the control. But with at 2 to 4 weeks with carbon tetrachloride, there's actually an increase with long and thin strings. And for all 4 characteristics, by 6 weeks, all 4 of the characteristics have increased. and with a reduction with drugs here. So now we're going to reticulation nodes, and just to refresh your memory, the reticulation nodes is when two collagen fiber cross. The point that it cross is the reticulation node. The more the collagen and the more complex the collagen network, the more the reticulation nodes. So this is our graph. So this is total, and then segment into portal, septal, and perisinusoidal. For total, you see that there's elevation in number of reticulation node at 2 and 4 weeks, with a peak at 6 weeks. Maybe a slight reduction with drug. For portal, there's an increase at 2 and 6 weeks, and it maintains that with drug. For septal area, you see an increase from 2 to 6 weeks and peak at 6 weeks with a reduction at with drugs. For perisinusoidal area, there's a visible elevation at 6 weeks with a slight reduction with drug. So this concludes the results for this study. Now I want to go into the ad hoc project that we did. So the next 2 slides will be the ad hoc project. As part of our innovative efforts, we want to look at the anti-LOX, anti-cross-linking activity. So we team up with Yuri Popov at Harvard. So we imaged the same exact samples that he published in his paper on the right. So this is the Pico-Cyrus red stain images that he generated. And then on the left is our image, the second harmonic generation 2PE image. You'll see that the septal bridge that form is very similar to his. But in addition, we see the bright red hepatocytes along the septal bridge that was not captured in his study. And also, we also wanted to add to our quantification parameters by enriching his study, such as measuring the changes of the septal bridge that you see in this slide. So on the bottom here is his image. He wants to measure the width of the fibrotic septal widening and collagen fiber splitting. So here's his measurement. So on the top here is our image. We see a very similar thing. We saw the fibrotic bridge splitting and widening. So we're developing a more sophisticated way to measure this fibrotic septal widening and collagen fiber splitting. Okay, so that concludes that. So we're now going to the second case study. So this is the fatty diet-induced liver fibrosis study in mouse. So I just want to show some of the images and the collagen quantification values. So what you see here on the top corner here is the normal mouse with lean diet, and then on the right, and these 3 images are from fatty diet-induced fibrosis. You see low fibrosis here, high fibrosis, and very high. So the green color you see here are the collagen, and what you really see is the collagen deposition elevation, and they are really collagen fiber in the perisinusoidal area. And I also want to point out the dark black circles here. The dark black circle here is actually the fat vacuoles. Fat vacuoles, they don't autofluoresce, so it shows up in our image as dark circles. So this is some of the typical value that you can expect when you measure a total percentage collagen area. Normal will be around 1, and then it can peak around to 13% collagen for images for something like this. So this is a very small area out of the whole tissue. This is about 0.6 by 0.4 millimeter out of the whole tissue, which is about 8 by 8 millimeter. Okay, so now we're going to a collagen segmentation for the images. So on the left here is the raw image, and then so now you see the total collagen here. The total collagen is segmented into the portal, septal, and parasinusoidal. So as we did previously with the carbon tetrachloride study, we can do collagen segmentation for NASH study, and in this case, fatty diet-induced liver fibrosis. In the portal region, you see the collagen around the boundary of the portal. Here is segmented very nicely. And then in the septal area, septal, what we call septal is the collagen between portals and the central vein. You see that there's a lot of collagen between the portal. And then parasinusoidal is the collagen in the parasinusoidal area that's not segmented into portal nor the septal. And they're usually very small collagen fiber, and you have to zoom in to be able to see that. And this is a zoom in version. And then, so for an image like this, you can expect about, to quantitate about 7% total percentage collagen, and then segmentation about 1% for the portal, about 5% for the septal, and then about 1% for the parasinusoidal. Okay, so now I'll bring the presentation back to Matt.
Thank you so much, Leah. I'm sure we'll have opportunity for Q&As after my last two slides. So we wanted to give you an overview of the capabilities and an example of very specific studies that we do. As I said, this technology is very, very new, yet well validated. And if any of you or your teams have interest to explore what we could do for your studies, I just wanted to highlight the way we develop the projects. We spend a lot of time to develop a study plan and then a resulting quote. All the work we do to develop a study plan is done for free, because every project is very different, and we want to make sure that the most of our technology is applied to your projects. Then we receive the slides. As Lee said, they would be very regular FFPE 5-micron tissue slides, non-stained. We process them quickly by batch. Usually if we want to increase throughputs, we would put 2 or 3 tissues per slide. So in one batch we could image 30 tissues, and we can do that overnight. And then the quantification comes, and Lee explained that we have 2 levels of quantification: the basic quantification level and the advanced quantification level. Everyone can start with imaging. Once you have the images, you can always decide to do the quantification later. The image are digitally stored. Our sponsors have those images. Usually they can start with basic quantification, and if there is any interest, we can retrospectively do advanced quantifications. And just to give you an idea, for a 20-slide study, it would take us all combined around 2 weeks, and the budgets would be between 20K or 30K, depending again on the level of quantification and if there are multiple tissues per slides. Of course, these numbers can be adjusted when there are bigger projects around. All right. So this is our workflow, and in conclusion, and that would be the last slide that I could leave here, Lee, if you don't mind. Yes, I wanted to summarize. So this is a busy chart, but let me talk through it quickly. We have explained that nonlinear microscopy, and in particular the technology that comes from our partner HistoIndex in Singapore, can be tuned to specifically image collagen and tissue context using second harmonic generation and 2-photon intrinsic fluorescence. The system, as opposed to a research instrument, is a commercial system for research-only use. It's a mid-throughput, non-destructive system optimized for conventional pathology slides. We can do imaging on all kinds of tissues, and we have experience on those, but what really matters is the quantification of those images. And right now we have three commercial solutions. We are very well aware that every animal model is slightly different, and we sometimes tune our quantification systems with the help of pathologists. Human algorithms are much more stable. And when it comes to translate those features into benefits, what we hear from our partners is, first of all, the robustness of the method, because we eliminate staining and reading variability. The fact that it's fully translational. Every protocol that you would do on an animal tissue is fully documented with metadata. So we keep track of all the parameters used for imaging and for quantification. If you do a clinical study, we would do the same on your human tissues, and down the road you can reproduce those tissues. Same thing, parameters used for animal images can be translated all along the development stage of your drug and applied again on human tissues. They compare the method and the outcomes compare with conventional staining method, but in addition, once you have done imaging, you have access to multiple novel fibrosis outcomes, either per tissue region, as Liz explained, portal, septal, or structural collagen, or fibrillar collagen, or you can really focus on other aspects of the fibers, you know, if they are the numbers of fibers we have, their shape, morphological parameters, but also the characteristics of the network and how those fibers get organized in the tissues, and in particular in the septal area. So these advanced quantification techniques are ideal for investigational studies. We are extremely sensitive of the progression of collagen. So we'll be happy to show you how we can detect signals in 2 weeks instead of 4 weeks, so we can reduce the time it takes to get a signal. Or we'll be happy to show you how we can detect very, very small effects of antifibrotic compounds. So the sensitivity is a big deal here. Of course, as you develop drugs and as you want to document effects into regulatory dossiers, there is always the opportunity to do retrospective studies. We're happy to receive a batch of tissues, retrospectively analyze them, and enrich the quantification of the impact of a drug that you would like to put in a submission, for instance. And we have been able to show that our turnaround time compares or outperforms pathology workflows. Everything depends on how you are organized. Some teams have limited pathology resources, so we become an overflow for very routine studies, while they use their pathology teams to do advanced projects, or because we are more competitive to pathology teams in the way of handling the workflows. Our system is fully automated, and all the image acquisitions can happen as we do quantification. We work in parallel. And last but not least, the tissues are not altered. We are totally non-destructive. They will be returned to you for any kind of additional study, biochemistry, for instance. So that concludes our presentation. I hope we were clear. We'll be happy to take questions. And Joanne, maybe you can moderate the Q&A session.
Well, thank you very much, Dr. Petitjean. That was a terrific presentation for you and Dr. Chen. We do have some questions. I encourage everyone to enter them in the question box on the right-hand side. I'll start with a couple that we've received. Has Genesis been involved in developing biomarkers for drug development that is some specific measure for example, safety or toxicology?
Lee, you want to ask? No, let me answer that. No, the answer is no. We acknowledge that this is an area of interest, but we have not done such. We are really focusing on fibrosis for now.
And then there's actually a follow-up question. So are you, which is asking, likewise, has Genesis developed any markers for efficacy of a drug therapy on treating a fibrotic disease? So it's almost the same situation, almost the same question.
The first one is really toxicity. The second question is more of an outcome addressing fibrosis, and the answer is yes. So I think some of the publications that I showed before would be the reference to support that answer.
Thank you. I'd like to hear more about the process of training the software, the analytical tools. Can you talk more about that process and where the inputs come for that?
Mm-hmm. Yes, that's a great question. Lee, would you like to talk about that, or do you want me to talk about it?
I think I'll have you talk about it.
All right, very good. So maybe you can show an image. Can you go back and show one of the image where there is segmentation?
Segmentation. Okay.
Yeah, one of the images, yes, what you have here. The 25 is probably a good image. Right. Okay, yes, I'm going to use that as a way to answer the question. So indeed, that's the area of most focus of what we do. So as I said, we process every tissue separately, and that's why we have the funnel of software development. So in an image that we collect from Genesis, there are two optical methods, the red, autofluorescence, and the green, which is collagen. Right? So for instance, in the liver tissues, the question is, how do we tell the software which is a portal area, septal area, and perisinusoidal area? It might be obvious for some of you, but in fact, pathologists are a great help to teach the software. Now, machine-based learning software are not new. These are technologies that are available. And basically what we do, we start with those hypotheses, and we would configure the raw features of the software to recognize our areas of interest. Then we're going to ask pathologists to basically flag portal areas. So pathologists are going to work on the image that is to your left, and with a pen or a mouse, really highlight the portal areas. And they're going to tell the software, listen, this area is a portal area, this area, and they're going to basically show 30 to 50 different portal areas on 30 different images. The software is going to learn from that. They're going to do the same for the septal areas with a different pen, or I should say with a different color, and by difference, we'll have the perisinusoidal area. So the pathologists will show that to the software. And that's what we call the development of our algorithm, right? Mm-hmm. So these softwares know how to do segmentation. There is additional thresholding, and every pixel that the software treats comes out with a probability to be either in the portal area, septal area, perisinusoidal area, and selecting the probabilistic level is basically one of the things we do with the pathologist in the validation phase. the validation phase is typically a project by which we compare, a sample of tissues treated by the machine. We show them to the pathologist, and we ask the pathologist to validate what they see. Right. So this is the protocol that we use to develop those algorithms. Now, I need to mention as well that what we have learned is that tissue preparation is a big deal. Sometimes the algorithm is tricked by artifacts that we have on the tissues, either slicing artifact or preparation artifacts or cleaning up the vessels from blood. So tissue preparation is a big deal and will very, very much help the segmentation. If needed, we can do a digital cleanup of the tissues, but it's a word of caution that we already discuss as we develop jointly a protocol to do advanced segmentation and quantification.
Thank you. So I have kind of a two-part question, and I think some pieces of this have been covered, but it would be helpful to hear some more. So we'd like to, it's basically about the advantage of working with non-stained samples. And the second part of the question is, what other detection methods have you compared this with? And I saw that you did say that it compares well, but can you give a little more detail about other methods that you have looked at?
So the first is non-stained tissues. Yes, so we take tissues, paraffin blocks, for instance, and those paraffin blocks are sliced. We don't do the slicing. Your teams usually do it, and they are mounted on glass slides. Well, the advantage is that we avoid staining artifacts, and we expedite the process, right? The other advantage is that after we've imaged the tissues, you can really reuse the same tissues for additional studies. We can also work with frozen section tissues, and that's very relevant when we do, patient studies. So that will work as well. Now, what have we compared that? That's a good question. I'm going to try to make a short answer and refer to some of the papers we presented. Of course, we have compared. The basic comparison is against the conventional pathology assessment. So we split our cohorts into two groups. One goes through our imaging and quantification workflow, and that would be for a human study that is referred in one of the papers I shown before. And the other arm of the study goes through the conventional pathology. And in conventional pathology, usually you have a pathologist scoring system, so it's a manual scoring system. We have also worked with sponsors in the pharma research that have used automated scoring system, right? At this level, both technologies compare well. We have a greater dynamic range and a much more sensitivity at early fibrosis levels. In addition to that, our technique has been benchmarked, and this is the Q Fibrosis paper that presents that very well, has been benchmarked to any other clinical assessment of liver conditions. So you have the entire liver panel that has been correlated to our advanced outcome methods, and we are currently working to enrich or correlate our advanced imaging techniques with advanced biochemical and other fibrosis markers. Just to conclude, I want to say that we are specific to collagen 1 and 3, so we can't compare to collagen 4 biomarkers, for instance. I tried to make it short, but it's a very good question.
Oh, that's terrific. It's been a great discussion. Thank you. Before I ask you both for final comments, I just want to let everyone know, and we'll be following up with you, that we will have on June 8th at 11 a.m. Eastern, we will be having another program on applications for lung tissue. And so I think that'll be a very interesting one as well. Dr. Chen, would you like to make some final comments?
No, it's just thank you for joining us, and we appreciate your attendance.
Thank you. And Dr. Petterjohn, do you have anything final you'd like to say to wind us up here?
No, many thanks. No. First of all, many thanks to you and your team and the Biopharma Research Council for making this presentation possible. And our contact details are probably somewhere on the website if any of you want to follow up, or if you have requests for some of the publications that we talked about.
Beautiful. We'll be sending out an email with the link for the recording, and we can include your contact information in that. Well, have a lovely day, everyone, and thank you so much. Take care.